All right, we're going to try to move a little quicker. Not getting very far from slide to slide, from video to video. But so here, active transport. So this is, now we're going to require energy, specifically ATP, right? So this is the energy molecule, the only form we really are evolved to use. So in this case, right, energy is needed. You need a carrier or a pump still, right, to get it across. And it generally moves against a concentration gradient. So generally, we have something that is in a place there's little of it, and we need to move it someplace where there's a lot of it, from low to high. Sodium and potassium ions are good examples of this, and this happens every time. Again, here shows the concentration. And this is a good thing to get out of the way right now, right? That sodium is supposed to be really high outside the cell, and potassium is supposed to be really high inside the cell, and therefore low, right, outside. And sodium is supposed to be low inside. Well, after depolarization has occurred and some sodium has moved in, but not the whole, all of it, right? We have to push, right? Use active transport to push or pump the sodium that came in back out, restoring, right, a resting membrane potential and homeostasis. And the same thing happens for the potassiums. The potassiums had been inside and they went out briefly. Well, we've got to put them back in. So we talk about these sodium-potassium pumps, all right? Here's another example, right? And this is if you are just too big for a carrier or channel, and it does require energy, right? So it's called endocytosis. It's like cell consuming. In fact, phagocytosis, cell eating, is one example of this, all right? So in this cartoon, again, we have a fluid, some solution that has large molecules. We have a solid particulate matter, right? And then we have what we call a receptor-mediated endocytosis. In each of these situations, you basically have the cell membrane surrounding this larger molecule, or molecules, and then surrounding it entirely and actually coming together and fusing to create a vesicle. So it looks like, you know, over time you are eating this particle. And once it fuses, then the whatever the molecules are now inside, right, the cytoplasm. So this is a consuming, whether it's pinocytosis, phagocytosis, or receptor-mediated endocytosis. And you don't, I'm not going to require you to tell me about the three types of endocytosis, but I do want to introduce them to you, and you will read about them. All right, so requires energy, the substance is too large, right? You form a vesicle, part of the outer membrane binds a substance and closes it and carries it into the cell, endo, inside. So you also have to have a way to get things out of the cells. So exo, exiting, all right, or exporting. Also active, requires ATP. Generally, again, something large that I don't have a carrier for. And these are all membrane-bound, right, organelles. And then here's the ribosomes that don't have a membrane. So here we see some of these membranes pinching off themselves, forming, right, vesicles, and the vesicle is carrying something this cell made, and we want to release it for something else in the body. And so this process, right, from here to here, fusing and releasing, called exocytosis, also known as secretion. And this, of course, is showing a number of different these little vesicles full of some sort of secretion. This is the pancreas. So these are probably enzymes. So digestive enzymes by pancreas, mucus by salivary glands, breast milk from mammary glands. Neurotransmitters are also exocytosed in the process of stimulating neurons, one neuron over the other. Okay? All right, so that's our plasma membrane, and we've really, really hopefully exhausted what we need to know at this level about the plasma membrane. So what else is required to be a cell? Your cytoplasm. It's known as the cytosol, and of course it, for us, contains organelles, and the majority of these are membrane-bound organelles. And I said this is specifically where we can get 290, really more, much more, different types. cells vary in what they need, right? So the numbers and types of organelles are different from cell to cell, right? And that's because there's, differences in what the job of the cell is. And this is just a nice generic cell to show you kind of the whole gamut. For the most part, these are represented on your cell model in lab, though you can't really tell the difference between a peroxisome and a lysosome on that model. But that's okay. Well, we'll still talk about them. And it doesn't really show the microtubules except with the centrosome. So, all right, so here, as we talk about these organelles, we look at a very, very important grouping of organelles called the endomembrane system. So within the membrane system. And these are generally also, again, most organelles are membrane bound with the exception of ribosomes shown here. But ribosomes often associate with a membrane bound organelle called the endoplasmic reticulum, right? Now, if those ribosomes are associated like they are here and in this cartoon, then we call it rough endoplasmic reticulum, right? And because ribosomes excel at forming the primary structure of proteins, right? They basically have the machinery to put the amino acids together in the right order, right? We know that these would be sites for protein synthesis. And what happens is as the ribosome builds the protein amino acid by amino acid by amino acid, then it enters into the endoplasmic reticulum, and inside here are a series of chemical reactions that allow for the folding of that protein into its secondary and sometimes tertiary structures. Now, if there are not ribosomes associated with the endoplasmic reticulum, it is then called smooth, and it does not participate in protein synthesis because it's the ribosomes that are really important here. And we say that smooth endoplasmic reticulum is important for lipid synthesis, for storage, and for detoxification. Okay, so here again revisits the ribosomes, the builders of proteins, and proteins, remember, from last week, or chapter 2, are all about every other function but energy, really, because energy is covered by carbohydrates and lipids. So the ribosomes actually form in the nucleus, and they are made as pieces, we call them subunits, and they would be exported out. And they are going to, again, they have ribosomal RNA associated with them that acts like an enzyme, and they basically bind to messenger RNA, and they have a binding for transfer RNA that we're going to look at later. And all these components get together out in the cytoplasm, and again, it just builds the primary structure, amino acid after amino acid after amino acid, of proteins. The unique thing about them, again, is that they do not have an actual membrane. Okay, so this blue here is supposed to be a membrane. All right, so some molecules require modifications. So some of what the cell builds is going to need to have some other chemistry attached to it, and or signaling molecules. So to get that done, it turns out that it has to go to another organelle. So, and I say the Golgi apparatus really does modifications, chemical adjustments. So here again, it is in an ER, right? Excuse me, ER. Sorry, electron micrograph, right? From an electron microscope. And this shows, again, it looks like a stack of flattened pancakes. There's actually, these are tubes that are interconnected. We've just cut them here in that picture, and here we've sectioned them in the cell. But here's the way it works. So this protein, in this case, this is protein synthesis because it's rough ER, has moved all the way through these interconnected tubes. It gets to the end, and we're going to pinch off this little vesicle containing our protein that we're trying to work on and build and optimize, right? And it'll move to the Golgi apparatus, and it will fuse. This membrane will fuse with this membrane, and it'll begin moving through here, and within the Golgi, it will be chemically altered. And so we say the Golgi refines the protein, packages, distributes, and allows them their distribution through signaling compounds. And so they could, if it's a protein coming from a rough ER, it can do that. If it's a lipid from a smooth ER, Golgi can still participate in these chemical adjustments to these large macromolecules. So we find Golgi more often in cells that are very good at secretion. All right, because those chemicals are due to be exported, and so they need to be modified so that they end up where they need to go, right? And I give you examples of like the pancreas and the salivary glands their cells would have lots of Golgi. All right, vesicles, ways to contain, control, export, or import. Okay, so I've mentioned this word a couple times, so it's a very generic word. I usually call them shipping containers, right, as I said here, right? And they transport or store, so they can stay inside the cell. So lysosomes and peroxisomes are actually a type of vesicle that stayed within the cell. So, but if they move to the cell surface, they can fuse, as we see here in this cartoon, and release their molecules. And we call this whole process secretion. These vesicles will pinch off either from Golgi or ER. And like I said, if they stay within, we call them a lysosome or a peroxisome, right? I say, when you talk about lysosomes as a type of vesicle, they formed from the Golgi, so this could be a lysosome. They're not trying to represent that here, but it could be. And they contain about 43 different enzymes that will help digest and recycle. So break down and recycle worn out organelles and chemicals inside the cell, as well as, in some cases, it can recycle the components of something the cell has consumed, like a bacterium, right? Right? So I say here, we've covered the assembly and recycling or defense role of the factory, because cells are definitely factories. Where does all the power come from? And of course, that's this modified organelle called the mitochondria. And I do remind you, this is why you breathe, right? And this is where, of course, ATP is made. It's not a very high resolution picture, but anyway, right? The very simple molecule with the adenine and the ribose, but additional phosphates, right? And very unstable, because all these are negatively charged and they don't like to be very close together. So it tends to fall apart, and as it does, it releases its energy. So the energy that's holding it together. It's always described as kind of like a spring, a loaded spring. All right. So mitochondria are pretty profoundly organized. They're double membraned, have a lot of chemical reactions going on, and they go on in different places within the mitochondria. Mitochondria has its own DNA. Mitochondria is believed to be now an endosymbiote. In other words, it was a microbe or a single-celled organism, very small, that entered into our cells a long time ago, right? Again, lots of chemical reactions as well, but done basically to synthesize ATP. So this is really a primary function. That's why we talk about it as the energy organelle. And ATP, of course, is the only molecule that can get jobs done in the body, right? Bring about change. Cells can have a whole variety of numbers of these, but on average, again, there's about 1,700 in a cell, and it can be thousands depending on, especially when you're talking about something like cardiac muscle cells. All right. In the course of all these chemical reactions, toxic waste, and this is what we tested in lab, right? We looked at these peroxisomes, which contained the enzyme catalase, and looked at its specific role in breaking down hydrogen peroxide to oxygen and water. So peroxisomes have a fairly straightforward, so they're membrane bound as well, all right? And they are found in a number of cells, but they're very high in very high amounts in liver and kidney. They have not just catalase that we looked at and isolated from liver tissue that had a bunch of cells, and cells with these peroxisomes. The peroxisomes also have enzymes called peroxidases, and peroxidases actually are forming the peroxide as they're breaking apart, as I say here, detoxifying or breaking down lipids. Hydrogen peroxide is a byproduct, and so that hydrogen peroxide has to be broken down further, and so catalase and peroxidases would be found inside here, both enzymes, and so this is really a detoxification place, all right? And there's also, reactive oxygen species. This helps works for antioxidants as well. So Peroxisomes, again, are very important and vital to our abilities to maintain our cells.